Our next speaker is Robin Mansukhani from Deciduous Therapeutics, another one of our amazing sponsors, and we're so happy and pleased that you can make it, and we very much look forward to your talk.
Cool. Thank you. All right. Thanks so much to the organizers for having us present here. Don't tell the other conferences, but this is easily the most fun conference of the year. So it's just really wonderful to be amongst all of you and share a little bit about what we're doing. So the core belief at Deciduous is that a properly functioning immune system is one of the best ways to extend health span. More specifically with respect to our work, we know that a healthy immune system can regulate senescent cells as nature intended. The issue is that over time with disease and aging, that immune function that would normally remove your senescent cells gets compromised. But the good news is that with the right approaches, we can restore immune function, and as I'll show you in some of the data today, this will not only remove senescent cells, it also has an impact on certain diseases that could shift the standard of care in a very positive direction going forward. Okay. There are 5 parts to my talk today. First, I'm going to talk about senescence and how its accumulation is really a failure of the immune system. 2, I'm going to talk about how we identified and eventually can restore immune function back towards senolysis. 3, I'm going to talk about why any of this actually matters. So I think senescence is interesting. There's lots of papers on it. I think the immune system is very interesting. But at the end of the day, what you need is data that suggests you can change an indication in a paradigm-shifting manner. And so that's some of the data I have to share with you today. 4, I'm going to talk about the path forward, so where we are today and then where we're going in the next 18 months. And lastly, just a fun bonus. You know, at home in California, I have 3 kids, and I have this job, and there's like not a lot of time to think about anything else. But when I travel and I'm alone and I have jet lag, instead of trying to be overly productive, I just sit around and think about like just terrible, terrible jokes I can tell to large audiences like this. So I have one today. I've never shared with anybody before. It's probably a bad idea, but we're going to do it anyway. Okay? This is the question. So as I'm speaking, you need to think about the answer. I'm going to ask you for the answer at the end. Okay? Now I know what it is, obviously, but this is the question. Like, what does a senescent cell actually say to the immune system? Okay? So you can ignore me for the next 15 minutes, but I'm coming back to this at the end. Okay? You got a deal? Okay. All right. So first, the business. Okay. So first, senescent cells and their accumulation. So as many of us know, senescent cells are irreversibly damaged cells. They're in permanent cell cycle arrest. This can be enabled by glucose, ROS, telomere shortening, injuries, diets. There's a number of reasons that cells become senescent and end up going into permanent cell cycle arrest. And over time, what happens is these cells go, hey, look, I'm in permanent cell cycle arrest. They start eliciting cytokines, GDF15, PI1, TNF-alpha, TGF-beta as part of the SASP profile. And really the way that we see it is this is their call for help. They're trying to say, hey, I'm permanently damaged. It's time for you to remove me, immune system. I've had my party. I'm done. Nothing else I'm doing here is useful. Let me now get removed. The issues with disease and aging is that immune function that should remove these senescent cells fails. And over time, in layman's terms, what I'd say is these cells become contagious. They, through cell-to-cell contact, will make other cells in the microenvironment also become senescent. And basically, it's like their second lease on life. They're saying, look, if you're not going to get rid of me in a timely manner, I'm going to amplify the signal, I'm going to poison the well for other cells in the microenvironment, and I'm going to destroy tissue in that manner. So over time, you get inflammation, fibrosis, and stem cell exhaustion. Okay. These are indications where senescence is believed to be causal in human disease. There are also indications where, as referenced in the literature cited there, that when you use any kind of senolytic approach, whether it be the immune system or other senolytics that are out there that have been published on, that this by itself will resolve diseases in appropriate preclinical models. Also, as noted in the orange, these are indications of massive unmet need with massive markets. So it just gives you a roadmap of if you are successful in this space, and you have a good approach that's safe and targeted, where can you take it? And this just gives you a sense of how big the market opportunity can actually be. Okay, so how do we do this? The first thing we wanted to do in our program is we wanted to look at different types of tissue, right? So one is adipose tissue, so metabolic tissue, one is lung fibroblasts, the third being autoimmune tissue. And the idea was not just to get an approach or not figure out how the immune system regulated a subtype of senescent cells, but to look broadly. So to say metabolic, fibrotic, and autoimmune, is there a common thread there that we can pull with the immune system? So we looked at these cells, we collated the senescent cells from these non-adjacent indications. We did single-cell RNA sequencing to understand their genetic profiles and what mRNA profiles they had that we could pull on. And long story short, so it's a short talk, so I don't have time to give you all the details around all the clues that are there on the way, but if you want to read the paper, it's open access. It's available. We published about 2 years ago. And we talked about how we discovered that natural killer T cells can, in fact, regulate senescent cells in the way nature intended it for it to be. Okay, so high level what we're doing and then the data. So when you're young, you're healthy, your NKT cells are very active, keeps your senescent cells in check. As I mentioned, over time with disease and age, your NKT cells, they don't necessarily go away, but they get energetic. They basically become like parents. It's like, they know what's going on, but they're kind of like slow to react at times. They slow down a bit, and that allows for senescent cells to now accumulate. But the good news is, with small molecules that we've developed, we can restore NKT function, and we can remove senescent cells. And as I'll show you in a moment, this has a pretty big impact on disease. Okay, so now does it actually matter? So first model, this is an IPF model. So first I'll explain the model, and I'll take you through the data and the target engagement. This is an IPF model, so bleomycin injury through the trachea. It's a fairly acute model, so you'll see on day 0, we do the injury to the lungs. In the first 7 days or so are the inflammatory period. You get inflammation, and eventually fibrosis will then start to take over in the lung after day 7. And then it'll go on for about another week or so. And our goal here is to not treat too early, because that's not realistic for human disease. Let's treat somewhere in the middle of disease. So when fibrosis has already started to plateau, can we give a drug then and see the impact on fibrosis and actual functional endpoints? So at day 10, during the fibrotic phase, we give a single dose of this drug. So it's 1 dose for the entire study. And the first thing we want to see is, did we actually hit the target and restore NKT cell function? And so as you see in the black are the age-matched controls. In the green are the injured animals that have not been given the drug. And in the orange is our drug. So now we know we have now expanded the NKT cells in the lung. We've now reduced the senescent cells as a function of activating NKT cells back to homeostasis. So again, the orange is a treatment group. We now see them returning back to the control levels in terms of senescence volume in the lung tissue itself. Okay, now, does it actually matter? Okay, so again, same model, just for your reference at the top, single dose at day 10, and at day 21. So at day 14, I showed you that we reduced senescent cells. Does it now impact inflammation and fibrosis? So these are inflammatory markers. TNF-alpha has been restored back to homeostasis, and then IL-17 has also been attenuated. And these are interesting cytokines because we know these are pre-fibrotic cytokines. So if you're affecting TNF-alpha, if you're affecting IL-17A, you'll also see an impact, ideally, on fibrosis, which is the second endpoint. And so we see here with a single dose, by day 21, you get about a 75 to 80% reduction in fibrosis. So again, the paradigm shift here, we've only given this drug once, but it can enable a massive reduction in fibrosis. The current standard of care with nintedanib, you'd give it about 10 times in this model at about 100 mg per kg per day. In best case, you're going to see about a 20 to 30% reduction in fibrosis. And the reason is because senescent cells are very pleiotropic. They give off many cytokines, and so if you're hitting a very pleiotropic target, it can have a much greater effect than just trying to go after singular targets in this disease. Okay. So I think I know some of the questions that are coming, so I'm just going to answer them right now. Yes, we are going to continue to evaluate this in older animals. We've gone as high as 18 months in our first pilot study and showed very good effects. We now want to keep pushing that up a little bit higher. The second pilot study we have ongoing is to look at functional endpoints beyond just fibrosis, but does it change oxygen saturation or lung function? The first set of data we have back on that is also very promising. We're just looking now and repeating it. Third one also is just doing the head-to-head. So based on the literature, we think nintedanib is going to be about a third as effective as our approach, but now we want to go show that in a single study and show that a single dose of our drug could be equal to or better than nintedanib in fibrosis measurements. Okay, so that was one. That's an IPF model. The second model we work in is now high-fat diet, so basically metabolic disease. Much longer model this time. It's not acute. This is very chronic. So these mice get a 120-day diet before we go in with any kind of treatment. During that 120 days, they become obese. They have elevated HbA1c. Their insulin resistance is very high. They're glucose intolerant. And so the question is, after all that happens, can we dial it back? So day 120, again, we go in with our single dose of the drug. Again, target engagement, NKT activation in the adipose tissue. Senescence reduction, again, in the adipose tissue. So again, the standard here in the black is the age-matched control, so not given the high-fat diet. The green is the high-fat diet animals without our treatment, the vehicle, and then the orange is our treatment group. Okay, so again now, what's the impact on actual disease endpoints? Okay, so again, model at the top for your reference. We look again 10 days after the single dose of the drug. We see now the mice are far less insulin resistant. And number two, when you challenge the mice with a bolus of glucose, they can now metabolize that far more effectively. There's a lot of news around Wegovy and Ozempic. I want to say this approach is not driven by massive weight loss. We see a very, very minor weight loss with these animals. It's really not driven by the mice eating less or somehow slowing down the stomach emptying and increasing leptin levels. This is driven by removing senescent cells in the adipose tissue, which normalizes the immune system in the pancreas and allows it to more effectively monitor and actually keep your glucose in control. So it's a different approach than what you're hearing about right now, and we think that's a good thing. Okay. On the long term, we also look at HbA1c. So HbA1c levels are historically a really good enrollment criteria for metabolic studies. Also, when you go into a doctor and get tested for diabetes, HbA1c tends to be one of the key markers that you look for to know if you're diabetic or pre-diabetic or if a drug is working or a lifestyle change is working. So again, same model, 120 days of diet, single dose at that time point. What's interesting here is that we see over a 60-day effect. So in the green are the high-fat diet animals that have not been given the drug, and over time, as you'd expect, their HbA1c levels continue to climb. In the black is the control animals, so the healthy animals that are just baseline, and the purples are treatment group, and they've only been given one dose of this drug at day 120. But I should also mention they stay on the high-fat diet through the entire study. So all the way to day 176, they're on the high-fat diet. And during that time, HbA1c levels are actually coming down as a function of this approach. Part of the reason is because it takes senescent cells a long time to accumulate. So if you remove them on day 20, you're not going to see the effects start to rebound until about day 80 or 90 with this approach. So we envision going forward, this is something you do about once every quarter. Senescent cells, when you remove them effectively, they don't repopulate very quickly. So that's the idea. So this shows about a 60-day effect. Like I said, it'll start to pick back up around day 85. Okay. So what are the advantages of this kind of approach? Single dose efficacy, everything I showed you today, it's one dose of our drug that has that level of impact. Systemic, so you think about aging, you think about age-related diseases, you typically don't have one single disease, you typically have multiple diseases. This is a systemic way to ablate senescent cells, which is really interesting in a disease like metabolic disease, which is really a basket of lung, liver, and kidney and cardio issues. It's a natural mechanism, so it's the way nature intended. Like I said, I think aging and longevity really should always hinge upon what's the way it was supposed to be and try to rejuvenate that first before trying other approaches that are more obtuse. Good precedent for safety, both in the NKT activation mechanism, also with our own drug. We just completed a safety study in rats where we showed up to 30 times the effective dose, no significant adverse events. It's robust, so it didn't matter in this case how we induce senescence. It could be through injury, it could be acute, it could be chronic through diet, but the effect from the single dose of our drug was still the same. Really nice cost of goods here. So we're talking about intermittent dosing, so it's a very different treatment paradigm versus other diabetes medicines, or even like in the IPF space where you're taking several pills a day that have poor compliance. This approach would suggest you can do this once every few months and still have a massive impact on the endpoints. Okay. So we're doing going forward. We have our lead compound, our formulation, and route have been selected. The CMC portion has been de-risked. We've made progress to the IND in terms of completing PK studies, TK studies, and dose-ranging safety studies up to 30 times the effective dose with no significant adverse events. Next critical path is scale up the compound and do a large animal safety study. And we're also now expanding this work into the brain. So not just in the periphery, but now we're thinking about brain aging as well. Okay. Many thanks. We've got a couple pharma sponsors, Golden Ticket non-dilutive awards from AbbVie and Eisai. So thank you to them. And of course, most importantly, I'm coming back to the critical question. Any volunteers?
Look, don't touch.
That's a good one. Look, don't touch. I give it a 4 out of 10. Okay. All right. We won't use that one again. All right. There you go. All right. There you go. You try. Okay. If any of this work is if you're a scientist trying to get into your industry, or if you're in industry and want to go from big company to small company, we're hiring actively in San Francisco on the formulation side, immunology side, and on the neuro side. So if that's of interest to you, or if you want more jokes, my email, not my email, but the general email is up there. Feel free to reach out, and be happy to talk to you about any opportunities. Okay, thank you guys so much.
All right. Thank you so much, Robin. That was really very interesting. Nia is jumping out of his seat, I can see.
Thank you very much. This is incredible. I remember last year, I thought it was great, such a good progress. 2 quick questions. IPF, what you did is not really the model we're all doing for IPF. Why have you done a tracheal injection, and is this really the gold standard model? I wonder. The second thing, by the way, for the diabetes-related issue, you didn't show the pancreas, but what I'm saying is you got senescent cells out of the adipose tissue. There's a lot of them probably, but also maybe around the hypothalamus, the pancreas. In other words, did you actually achieve a much more body homeostasis for glucose metabolism than only the adipose?
Sorry, I missed the last part of the question.
Did you get this more systemic effect than
yes.
suggesting it's only the adipose?
Yeah, so actually it's studies we're starting soon. We want to see now just is it a diabetes treatment, or can it be extended now into affecting the cardiac, renal, and liver markers? So it's actually, you're exactly in line with what we're thinking about right now. But those studies take a long time, and we're actually just started them. We started the diets on the older mice last week, so it's going to come out probably by the end of the year, early next year. We'll know, is it just this approach, how much further can it go in a normal metabolic diet? Sorry, so you're talking about probably the transplant model, or which one are you referring to?
No, doxycycline versus
Oh.
the tracheal.
Yeah, the intratracheal bleo. Yeah, I think at the end of the day, they all seem to have certain pros and certain cons, so we've elected to work in the bleo model first. Actually, the next step, so there are different ways to induce fibrosis in the lung in vivo. We actually think the next best step for us is to look at human ex vivo studies and look at actually IPF patient lungs and try to see a similar effect with actual human IPF patient lungs. So, I think you can spend a lot of time on preclinical models with IPF all day and probably not convince yourself too much this way or that way after you do one. So I think it's actually human ex vivo is actually the next step for us.
Yeah. Can you just take a second to explain sort of how the mechanism by which a single injection is resulting in this very long-term effect that you're seeing?
Yeah, so it's 2 things. So one is the single injection restores the NKT cells, it proliferates them. And the NKT cells have an endogenous ability to recognize and remove pathological senescent cells. It so happens that the proliferation we get from the NKT cells is enough to ablate a good proportion, as I showed, of the senescent cells. And so once you've removed them, the time it takes for them to repopulate and for cells to go from normal, healthy to senescent again, like in a high-fat diet model, takes a long time. It's not something that we did that's great. It's just that in senescence, these cells, like senescent cells, don't just populate in a day. They tend to take weeks, sometimes months at a time to actually come back. And not only do they come back, they have to come back and actually, again, be pathological and actually cause destruction. So you can, we all have like a low burden of, when you're healthy, you can have a low burden of senescent cells all the time. That's okay because your immune function kind of can equal them out. The issue is when they start to accumulate aggressively, you need to get them back down to baseline.
Thank you.
All right, thank you so much, Robin. Really fantastic work you're doing. And thank you for the support.